welcome to another biochemistry episode. This time we're talking about the citric acid cycle. For those of you who haven't watched my video about glycolysis, you can do it now because the citric acid cycle is basically the next step. Other names for the citric acid cycle are Krebs cycle or tricarboxylic acid cycle. And it's the second step in aerobic respiration, and therefore we've got glycolysis first, citric acid cycle, and then oxidative phosphorylation. This pathway plays crucial roles in various biochemical pathways. So this led to the conclusion that it's possible that it's one of the oldest parts of cellular metabolism at all. So, if you remember glycolysis, at the end we've got two pyruvates. So these pyruvates can be converted into various molecules. Among them is lactate, which is quite famous. The other one, and the relevant one for the citric acid cycle, is the Acetyl-CoA. The reaction from pyruvate to Acetyl-CoA is actually catalyzed by a complex, the so-called pyruvate dehydrogenase complex actually, and this is formed by three distinct enzymes, which are pyruvate dehydrogenase, and the other two are dihydrolipoyl transacetylase and another dehydrogenase. And as this reaction can take place in the cytoplasm, we have to transport pyruvate first into the mitochondrial matrix, which is where the whole process takes place, which is a contrast to glycolysis. In glycolysis, we said that this happens in the cytoplasm. Now we're in the mitochondria. Another difference between glycolysis and the citric acid cycle is, as the name suggests, as it's a cycle, we can repeat this pathway over and over again as the reactions itself regenerate the molecules that are required for the first reaction. And actually here there's nothing like a first reaction because it's a cycle. But let's stick with this. In total, we've got 8 different enzymes that catalyze the reactions. The first step itself is a 2-carbon acetyl group, which is transferred from acetyl-CoA to oxaloacetate, which then forms citrate, which is also called citric acid. So this is the molecule that is responsible, or that was used to name this pathway. This citric acid then basically follows the cycle where it undergoes various chemical transformations. We're not going to talk about them in detail, but you see it here. The intermediates itself that are formed during this process are also useful because they are used as precursors for synthesizing other molecules. Now, the main thing about this whole pathways are electrons. So, the vast majority of them is necessary to form NADH, which is going to be important in oxidative phosphorylation later on. We're going to talk about it in the next video. And we've got another electron carrier, which is called FADH2, which can be converted into ubiquinol, which is a substrate for the electron transport chain at level 3. We'll talk about that later again, as I said before. So both of them, so NADH and FADH2, are molecules that are necessary for generating energy in the inner mitochondrial membrane. Now you might wonder, within this cycle we produce just one ATP or GDP, which varies between different organisms, but this is the important step. We can use NADH, the 3 NADH and the 1 FADH2 that are produced during this cycle to generate even more or a lot of ATPs in comparison to glycolysis in oxidative phosphorylation. So as you might notice by now, this is important. So stay tuned and watch the next video.